Hi, good morning, everyone. I'm Filipa Rijo-Ferreira. We just started our lab at Berkeley. We're interested in circadian rhythms in parasitic diseases. And the one we're mostly focusing on is malaria. And that's because malaria has been very famous for its periodic fevers, fevers that come and go at certain times of the day. And that's actually all a consequence of the parasite replication cycle in the blood. So this is an amazing phenomenon. You can think about these unicellular parasites coordinated in their invasion of a red blood cell, and then they replicate, and then they burst the red blood cells and release these millions of parasites as metabolic waste that then lead to the fevers. And interestingly, different Plasmodium species have different lengths of these replication cycles. Some take 24 hours, some 48 hours, and some 72. So all multiple of 24. And that really piqued my interest by the aspect of us in this world that have been really exposed to these very rhythmic environmental changes with a 24-hour light cycle. And in particular, different organisms have evolved ways to anticipate that. And so the big question was whether parasites also have, similar like other organisms, really evolved an intrinsic way to anticipate it, whether they have circadian rhythms. So we've tried many different ways to test that, and here I have just one experiment that really can show that once we get rid of the host environmental exposures and we get rid of the host functional clock by genetically manipulating these mice, we infect them with parasites, and the rhythms that we have very usually observed still remain. So really showing that the parasites themselves have an intrinsic way to tell time. And that kind of changed the perspective that we have now about this infection. So there's 3 different organisms involved in that being the host, the vector to transmit it, and the parasite, and now all of them have their own intrinsic ways to tell time, and they are very rhythmic in their biology. So this really raises many questions that we are currently trying to address. We are very interested in trying to figure out how are the parasites sensing their environment, what are the host cues, what are their environmental cues, similar, trying to understand how we tell time. And exactly how mechanistically does this work? So we're taking advantage of forward genetic screens and other more guided approaches to really find out that molecular machinery of what is regulating the parasite transcriptomics. And we're also starting a new venture, which is to really consider now the mosquito themselves and how these might impact transmission. Mosquitoes of Anopheles really bite at night. Now the parasite has a rhythm, the host has a rhythm of being potentially more susceptible as well. And we're trying to put this all in context to address how these 3 clocks co-evolved to establish this infection. Let me know if you have any questions.